Why hello beautiful people, Kendall Hailstone here. Thank you so much for joining me today. It is day 20 of my 90-day video challenge, and today's video I'll be highlighting a huge benefit of growing mushrooms, and that is learning about the life cycle of mushrooms. And so that's what this video is about, is the life cycle of mushrooms. So if that's of interest to you, please stick around. I'll be going into a fair amount of detail, which I'm really excited about. I just read some great information about it yesterday and did some research online about it as well. And anyways, so I have some great information for you today. If you are totally nerded and geeked out by the science of mushrooms, which I totally am, still definitely learning. By no means am I an expert, but I want to share what I've learned just because it's exciting to me, and I want to be making videos that I'm excited about. So yeah, let's go ahead and hop right into it. But first, don't forget to subscribe to my channel. Click that subscribe button down below to the right. Thank you so much. Give this video a big old thumbs up at the end if you feel like it was valuable to you. Share it with your buddies, share it with your pals. If you know anybody who's interested in mushrooms, you think they have that technical kind of science mind that they would find this fascinating, then go ahead and share that with them, this video with them too. So why don't we get right into it? Okay. First, I want to point out that fungus is actually more closely related biologically to animals than to plants, which I thought was fascinating. And so, for example, the cell membrane of the fungus, of fungi, is made of lipids, which is fat and protein cells. And then the cell walls are made of chitin, which is very similar to that of crabs, beetles, and other crustaceans. And so I just thought that was super interesting. And you know, whereas us humans and animals, we have a digestive system, you know, internally, where we, you know, put food into our bodies and we digest the foods internally. Fungus, which, you know, makes sense once I heard it and think about it, but I didn't really think about this before. Fungus actually have an external digestive system, and they, you know, excrete enzymes to digest their food. And these enzymes are called exoenzymes actually, and I just thought that was fascinating, which, like I said, makes sense. They digest their food externally, and then they absorb the nutrients through their bodies. It's fascinating. But anyway, in any case, so the life cycle of mushrooms, we're just going to start from a spore, and then we'll end at a spore, just to make it simple, just from the beginning to, you know, beginning, so to speak. And so let's get right into it. So first of all, I just want to point out that spores of most mushrooms, and there are exceptions to this, of course, which I'll point out, but spores of most mushrooms are genetically haploid and uninucleate, which means that they have one nucleus, which I'm sure you could get, nucleate. They have one nucleus per cell, and haploid means that instead of having a full set of chromosomes, they only have half this full set of chromosomes. So they need to mate with the sexually complementary hyphal mycelium network in order to be able to reproduce and make mushrooms. And mushrooms are essentially the fruiting body, or I guess you could say the sexual organ of the fungal body, the fungus, the mycelium, and it actually is what produces the seeds, you know, the spores. So in any case, let's go back to the spore. So a spore, it's haploid, uninucleate mostly, and it'll grow what's called a hypha. It's just like a one-cell strand of mycelium called a hypha, and it's also uninucleate and haploid, and it divides, and it creates branch-like figure, and that creates a hyphal network. And then once this hyphal network meets another sexually complementary hyphal network, and then it fuses, the cell walls between the hyphal networks dissolve, and they are able to exchange cytoplasm and genetic materials. And therefore the emanating, the secondary hyphal network or mycelium that emerges from that is now dikaryotic and binucleate. So the hyphal network that emerges after the exchange of cytoplasm and genetics has occurred, that is the secondary mycelium or hyphal network, and this is now dikaryotic and binucleate, meaning that each cell has 2 nuclei and that it carries a full set of chromosomes so it can now reproduce. So we are in good shape already so far. I also want to point out that this one mycelium that is dikaryotic and binucleate has 2 nuclei per cell and has a full set of chromosomes, and this other separate mycelium of a different species that is dikaryotic and binucleate, they can actually come together and mate to create a new strain. And that process of these 2 fully sexually mature myceliums coming together and fusing, making a new strain, is called anastomosis. I just thought that was really interesting, kind of, side note there. Okay, so anyways, so we have this secondary mycelium that has grown. Now it's capable of producing mushrooms. And so something that's very common in the mycelium just prior to making mushrooms is that each cell of the mycelium network will actually be dividing, creating more nuclei, and sometimes they can have up to 12 nuclei per cell. And it's, from what I understand, it's getting ready to divide, rapidly to be creating these incredible mushrooms. And we're not going to get into super detail about the actual formation of the mushrooms. So in any case, so the cells have, like I said, up to 12 nuclei, and then the cell walls are formed and splits up these nuclei, so that's back to like 2 nuclei per cell, and it's doing this, and it's, like I said, producing a lot more cells ready for this mushroom formation. So mushrooms are growing and the cells are dividing like crazy. Up to this point the mycelium has been growing vegetatively, which means it's just the mycelium network is growing and expanding, getting more nutrients, looking for nutrients and all that good stuff. And once the fruiting stage is when the mushrooms start coming up out of the ground, and this could also be called the tertiary mycelium. Let me take a step back. So the original hyphal network that was created from that one spore that was haploid and uninucleate, was the primary mycelium, and the secondary mycelium is the mycelium that emanates from the sexually complemented hyphal networks that mate, and that's called the secondary mycelium. And then the mushrooms coming up out of the ground, that could be called also the tertiary mycelium. Okay, so in any case, we have these mushrooms grown, and in the gills, and different mushrooms have different ways of dispersing their spores, but we're just going to talk about, for simplicity, the gilled mushroom, just for ease of reference. And so in the gills of the mushroom, these cells are formed called basidia, and these basidia play a critical role in the life cycle and the reproduction of the fungus. So the basidia are actually formed when the gills start maturing, and the basidia form not all at once, but they form in stages, so that not all the spores that are accessible to the mushrooms to disperse are dispersed all at once, and it goes through a progression of spore dispersion based on basidia maturing at different times. And so the cells that actually give rise to the basidia are binucleate and dikaryotic, and so it has 2 nuclei, and each nuclei is haploid, carrying half of the full set of chromosomes. And so there is a certain point in time in the basidia's maturity when the 2 nuclei in one cell of the basidia actually come together, fuse together, and create a diploid nuclei. So it's one nucleus that has the full set of chromosomes, and that process is called karyogamy, with the fusing of the 2 nuclei in the basidia cell. And soon after this process of karyogamy, of the fusing of the nuclei, creating a diploid nucleus that has the full set of chromosomes, the basidia goes through the process of meiosis, and typically will create 4 daughter cells that are all haploid and uninucleate. Alright, so I hope you're following me so far. Okay, so the basidia has created these 4 haploid daughter cells, and actually has these kind of branch-like structures that are created called stigmata. And this is where the the daughter cells will actually migrate up these stigmata structures, and just imagine kind of like this type of scenario, each of these being a stigmata. Just imagine there's only 4 and the haploid daughter cells are moving up the stigmata and will congregate, not congregate, but they'll end up at the tip of the stigmata, and that's where the spores are. And those daughter cells are essentially the spores, and they come onto the tips of the stigmata, ready to mature and ready to be projected onto the free air. Okay, so the process of actually how the spores are projected is really super fascinating. In the Growing Gourmet and Medicinal Mushrooms book by Paul Stamets, it actually doesn't give specific detail of how the spores are projected, because at the time of that book was written, it wasn't actually known how the process was carried through. But I looked up yesterday how what the process looks like, and I found an Australian government site that outlined the process, and I was just blown away. Okay, so the spore is, this is the stigmata on the basidium, and the daughter cells migrated up, and it's at the tip, and the spore is actually connected to the tip of the stigmata here by what's called an apiculus. And what the apiculus is, and I hope I'm pronouncing that correctly, but anyways, what the apiculus is essentially a kind of spike-like, a thorn-like protrusion from one of the tips of the spore, and it's connected to the stigmata, by this thorn-like protrusion. And it's called the stigmata apiculus boundary, and at this boundary it's actually a point of weakness, and as the spore matures, it becomes weaker and weaker, and I'll kind of further explain of why that's of importance. Okay, so as the spore is maturing, it actually secretes small amounts of sugar molecules at the apiculus stigmata boundary, and the water's inside of the mushroom. Imagine you're a tiny spore and you're inside the mushroom. You're inside basically of the gills, not like inside, but you're on the underside of the mushroom, and it's very humid in there, and so water condensation is captured by these small sugar molecules and starts to condense and create a water droplet. The little droplet of condensation that is formed at the junction between the stigmata and the apiculus is called the Buller's drop. B-U-L-L-E-R'(S), Buller's drop. And at the same time, there's actually a thin film of water that's formed over a large portion of the surface area of the spore. So the drop here, the Buller's drop, is growing in size, as there's still water vapor in its environment, so it's condensing more water into this little drop. And then once the drop grows to a certain size, and it can grow substantially, quite large in size compared to the spore itself. And a keynote to point out here is that before this Buller's drop was formed by the spore secreting sugar molecules, the center of mass on the spore is more or less centered, and that's something really important to identify right now, because that's a key point in how it is projected. Okay, so the center of mass is more or less in the center of the spore, and as this Buller's drop is growing in size, the center of mass of the spore is actually moving closer and closer to this junction between the stigmata and the apiculus. And so once the Buller's drop reaches a certain size, it actually will touch the surface of the spore, and the water drop will collapse, and the water will actually flow onto the surface area of the spore, which already had a thin film of water, but the water will flow onto the surface area of the spore, and by doing so, the center of mass of the spore will drastically change directions. So it was close to the junction here, but now it changes directions to the direction of where the water flowed, and that creates a thrusting force, and ejaculates the the spore into free space, which is then carried off into the air currents, and it can germinate. And I just think it's also really cool, not only that whole process of that projection of the spore, just based on the change of direction of the center of mass, but also it's also creating more moisture for the spore, as it goes off into this free space, into this God knows what type of environment it's going to be in, right? But it's actually providing itself with more moisture before it gets sent off to germinate. And that essentially is the end of the life cycle of the mushroom, is when it disperses its spores, it has completed its mission, and now it is up to we are back to the spore, like I said, which in the majority of the case is a uninucleate haploid cell, and that has to meet another sexually complementary uninucleate haploid cell in order to mate and be able to make a secondary mycelium and reproduce all over again. Now, I just want to point out that the process that I just outlined is a very generalized process, and it varies greatly based on the species, but this is how it is for the vast majority of mushrooms. Like I said, I'm no expert, but this is just what I've learned, and I just thought it was super fascinating. But I do want to point out one other example, that instead of the basidia, like I said, creating 4 daughter cells, that go through meiosis, and after the process of karyogamy, the fusion of the 2 nuclei to create this one diploid nucleus, and then going through meiosis to create 4 daughter cells. So in contrast to that, there is another mushroom, I think it's just called the button mushroom, and it's in the Agaricus species, where the basidium creates 2 binucleate dikaryotic cells. And so each spore is actually fully capable of producing fruiting bodies, and so that one spore would be projected off, and just with the same bolus drop, the whole center of mass change in direction, all that good stuff. So it would be projected off, and then it would land somewhere, germinate, and that resulting mycelium would be fully sexually mature and capable of producing mushrooms, and it wouldn't have to mate with another sexually complementary hyphal network in order to create mushrooms. So I just wanted to point that out. That there are lots of different ways that mushrooms go through this process, but I outlined the most common one. So, all right, that pretty much wraps it for today. I hope that was relatively easy for you to follow. I mean, it makes sense to me in my mind, but I know there's a lot of technical terms, and maybe there's some things in here that you don't completely understand. Like, for example, meiosis. I don't completely understand what meiosis is, but I've been feeling a drive to learn a little bit more about biology, so that I can understand a little bit more of the nitty gritty of the mushroom growing and the mushroom life cycle and such. So anyways, thank you so much for joining me today. I hope you found this interesting, at the very least entertaining. And I hope you have a wonderful rest of your day. Get out of your comfort zone. That's where growth happens, people. So go out there and expand yourself in new and exciting ways. And I'll see you tomorrow on day 21 of my 90-day video challenge. Hope you have a beautiful, beautiful day. I'll see you guys later. Peace.